My name is Emily. I'm a senior curator at UniProt, and I also work as liaison between the curation team and the web development team. And I'm joined by Aurelian, who is our website project lead, so helps develop the website here at UniProt. So we're going to do a bit of a hybrid webinar, which is new for us. We're going to talk a bit about the science, and I'm going to give you an idea of how the website displays proteomics data and how we bring proteomics data into UniProt. And then we're going to touch on some programmatic access and how you might be able to download some proteomics data for further analysis yourself and bring it into your own pipelines. Our learning outcomes are, we hope you understand our 3 different proteomics pipelines that we currently have running at the moment and where to find that data in our website. And then briefly, you have an understanding of where our proteomics data is programmatically and that you can download it at home after the webinar. So UniProt in general, this is kind of my usual slide. We're a comprehensive, high quality, always publicly accessible resource of protein sequence and function information. So this includes primary sequences and isoforms. We curate physiological protein function, which includes its subcellular location, the pathways and reactions it's involved in, interactions, involvement in disease and variants. And then we also annotate the actual sequence of the proteins with domains, any post-translational modifications, mutations, disease variants, and much more. We act as a repository for structural information, which includes AlphaFold and experimental 3D structures. And we also have data analysis tools for your use, such as BLAST and multiple sequence alignment. UniProt is a consortium made up of 3 parts. So we're from EMBL-EBI in Cambridge, UK, but we also have colleagues in PIR in America and SIB in Geneva, Switzerland. So today's webinar, briefly, the structure, we're going to be looking at the website and I'm going to be walking you through how to download a proteome, our 3 different proteomics data pipelines that integrates proteomics data into UniProt, and then I'll be touching on each pipeline individually. And then Aurelian is going to take over and we're going to look at programmatic access using Python, so including setting up your working environment, getting HPP peptides for your protein entry, and if you have some peptides, how do you find out which protein they match to? Now, we're going to be talking about Python today, but you can access UniProt using whatever coding language you want. And we also have a UI that facilitates that as well in our documentation section, which we'll be touching on later. So let's start. How to download a proteome. So we have a set of proteomes, which we've got quite a lot now, and we've also got an extensive list of reference proteomes. And on our homepage, you can find them under the little red box saying species proteomes, or also in our search box at the top, you can click proteomes and then put your species of interest in. You can open up a proteome homepage. So in this case, I'm displaying human. We're going to be talking a lot about human data today, but we do have organisms from every single kingdom and a range of species and strains as well. Each proteome has its own proteome ID, whether it's a reference proteome or not, and its number of entries. So that's the number of protein entries in that proteome. The gene count that refers to, and obviously the taxonomy again. And then we also have some statistics such as Busco score, and each proteome will have a brief description of that proteome and the species involved there. To download these proteomes, you can do it either chromosome-centric or you can hit the download button, and we have multiple options here. So this has changed in the last year as a result of user feedback. So you can now download just what you've selected in the table below, or you can download just our reviewed entries, which are curator annotated, or you can download all entries associated with this proteome, which is reviewed SwissProt or unreviewed Tremble entries. And you see there's differences in numbers here as well. And that will vary depending on the proteome. You can then also download additional sequence data. So this includes isoforms from SwissProt, so our reviewed entries. That will limit your download to a faster format. So have a think before you do your download, what entries you want to cover in your proteome. We also have a range of formats you can download in, and whether it's compressed or not, and then just click the download button. We can also generate a URL for the API as well if you want to download that programmatically. And then moving on to how we get our proteomics data. So currently we have 3 pipelines running, and they're all separate. They all have a common feature, the fact that we map to our reference proteomes. And I'll tell you what I mean by that in a minute. So our three pipelines are, this is our newest, which is a human proteome project. This is new this year. We have data coming in from them. We have the proteins API endpoint for that, which we'll touch on later. And that data gets mapped to a proteome, and if it's a perfect match, it comes into UniProt's website, into the proteins API where the data is stored programmatically, and also into the feature viewer where you can see that representative data as well. And I'll touch on that specifically in the next slide. Our second pipeline is the oldest pipeline that we have running, and that is our proteomics pipeline. It's a filtered list of peptides from mass spec repositories, including Peptide Atlas and proteomics-DB. These are all public repositories. It has its own endpoint in the proteins API, and again, the data is matched to our reference proteomes before it is imported into UniProt. If a peptide doesn't match, it isn't brought in, so it has to match the protein sequences and in silico peptides that we have in UniProt to be incorporated into the database. And then we also have the PTM containing pipeline. So this is data and work we've been doing with a consortium of collaborators, part of the PTM Exchange project. And this is looking at mass spec peptides that contain post-translational modifications. It has its own endpoint, and again, it is mapped to our reference proteomes before it is brought in to both the website, the proteins API, and of course, the feature viewer. So I'm going to talk through these in greater detail. These two are multiple species pipelines, whereas obviously the human proteome project is just for human data. So the human proteome project, this is new, and we've only just started bringing this data in. So the Human Proteome Project, for those that aren't familiar, they aim to provide mass spec evidence for the existence of every human protein. We take proteome exchange datasets that the teams associated with the HPP have deposited, and we reprocess them and score them in Peptide Atlas's trans-proteomic pipeline, and we get the data from Peptide Atlas. We then exact sequence match it to our reference proteome, the human in this case, and as long as there's an exact sequence match, that peptide is brought in with its associated evidence to the database. That then triggers an update protein existence value to 1. So you'll see at the very top of a protein entry, you'll see, say, the gene name, how many amino acids, and alongside that, it will say what evidence we have for that protein existing. So we can have it at mRNA level or just genomic, but when we've got proteomic data, we say evidence at protein level. So that is the aim of the HPP project, is to have that evidence at protein level for every human protein that exists. So all the entries will be updated with that. We also have the keyword proteomics identification added to every single human protein entry with that sort of data from the HPP. And then, of course, that data goes into the website, the Proteins API, and our feature viewer. Inside our feature viewer, today we're going to be talking nearly exclusively about the proteomics track. The proteomics track on the feature viewer. So the feature viewer is a string of amino acids from 1 to the other terminus of the protein. So you can zoom in on a particular area and see what features and domains and information we have for that particular string of amino acids for your protein. So we map the peptides to these specific residues on our feature viewer on our string of amino acids. So they look like bars like this. The Human Proteome Project has its own track, and if you click on one of the peptides, you get a little tooltip that tells you exactly what that peptide's information is that we have for it. So in this case, that peptide is unique to that protein, i.e. it doesn't map to any other protein. It's from residues 180 to 196. You have the peptide sequence, the proteome reference ID that it's mapped to, and also its sources. So this particular peptide has sources from Peptide Atlas and Massive, which are both contributors to the Human Proteome Project. So that's the HPP project. Oh, look, I've even done a better screenshot of that. So proteomics track, Human Proteome Project, and you see we've got your amino acid sequence here left to right, increasing in amino acid numbers, and then you click on the track and it gives you the data for that particular peptide. This peptide is nearer the N terminus in this case. You can access this data programmatically via the Proteins API through proteomics/HPP section. We'll talk about that later. But I put that in the slides for reference in case you want to go back and look. So this is our traditional proteomics data pipeline. We bring in peptides from mass spec repositories that are available to the public, and we exact sequence match them. So that is coming from the right over here, the proteomics-DB and peptide atlas. We exact sequence match them to a in silico digestion model of our reference proteomes. So we have a set of methodologies that we do for in silico digestion, and those resulting peptides we match against the peptides we get from the databases. And as long as they're an exact match, we again assign evidence at protein level and import into the database. So that particular track, see, I don't have a slide for this, that particular track is unique peptide. And you might see in some entries as well non-unique peptide, and that just refers to whether those peptides are unique to that particular protein. Unique meaning it doesn't map to any other peptide in that species. Non-unique meaning it can map to more than one. And again, that's downloadable from the Proteins API under proteomics/non-PTM. And then, of course, we come to the PTM data. So post-translational modification data from mass spec is brand new, pretty much. We've only had the data in for a year. It's grown exponentially in the last few months. It's part of a collaboration from the PTM Exchange project, and that's a collaboration with UniProt, Pride, Peptide Atlas, and the University of Liverpool in the UK. So we work on proteome exchange datasets, which are publicly available mass spec datasets. These datasets are enriched for PTMs, and then these datasets are filtered based on their methods, their enrichment methods, sometimes their treatment methods. We try not to bring any potential non-endogenous or non-endogenously possible PTMs into the database. So we don't usually allow chemical modifications of datasets before we process them. We also look at sample type, the robustness of the study overall, and their own mass spec pipeline that they've used in their analysis. And then we take those datasets and we reanalyze them using TPP. And then we look at modified site scoring, which means that we confirm whether that particular residue within that peptide is the modified site, and then we confidence score them. So that is all done at the University of Liverpool with our collaborators over there, which will mean that every single PTM that comes from this data will be assigned a gold, silver, or bronze criteria based on their false localization rate of that PTM on that peptide. So that data doesn't just come to us, it goes into the ptmexchange.org, it goes into Pride. Peptide Atlas have a build, we call it a build, for all these modification data sets that we have. And then, of course, we bring it into UniProt, and we do this based on both the PTM and the proteomics data. So you can look at a protein entry, and the data will be in there specific to a site or specific to the peptide, which I'll show you in a minute. I say we've had a bit of an exponential increase in data for the PTM Exchange project recently. So we have phosphorylation data currently available for rice, maize, yeast, mouse, T. brucei, and malaria. We also have simulation data for human. And then coming soon, which is what we're working on at the moment, we've got human phosphorylation data and human acetylation data, which is in the very early stages of analysis. So where can you find this data? You can find it in the protein entry page, which sets it apart from the other two pipelines. You can find it if you navigate to your favorite protein on the actual page. You can click PTM/processing. And in that section, you have an amino acid string in your feature graphic again with the PTMs as little kind of ticks on where their location where they are in the string. And then you can also filter in the table. So to filter for this type of data, you want modified residue large scale data, or you can filter by source, PTM Exchange. And that will give you a readout like this. It will tell you which position is modified, how it's modified. So these are phosphoserines. I think this is rice data we're currently looking at. And then you can see we've got the confidence scoring here, bronze and gold. And if you click on the evidence tag, which you can find throughout the database, which explains where all our data comes from. If you click on the evidence tag for the PTM exchange data, it will explain that this PTM is a gold or a silver, and then the evidence for that as well. So this will link out to proteome exchange and peptide atlas as well. So we have throughout the website, I'll mention this now because I don't have a screenshot of this later, but we have loads of little eyes, which mean information. So if you click on these eyes, it will tell you, it will snap to the help page that's most relevant for the section where you've clicked. So we have a whole help page to do with PTM and processing, and then we obviously have sections of that help page to do with this specific data as well. So if you ever want to know where the data is from, what the data even is, where else you can find it in UniProt, or maybe where all the sources and the methods we've used, you'll find that on the help page for that data. So where can you find the PTM exchange data in UniProt? You can find it on the entry page as I've just described, and then you can go to the feature viewer, and we have it in 2 sites on the feature viewer. So we've thought about this from a biologist perspective, and we know that some people are comfortable with proteomics and some people are not. So we presented it in 2 ways. We have a PTM track, and under that we have a modified residue large-scale data. And we have little triangles that are color-coded according to confidence scoring. So gold and bronze here again. And they sit at the residue on the amino acid string where they're present. So in this case, at residue 150. The tooltip tells you more information. So it's a phosphoserine, it's a bronze. And the evidence here, they're the PXDs from PRIDE and Proteome Exchange. So you can click them and it will take you to the datasets that provide that information. And they are the original datasets that we've then taken and reprocessed within the PTM Exchange Consortium. And then in the proteomics track, it has its own track under PTM containing peptides. So the peptide is mapped to the amino acid string. And if you click on the peptide, it tells you the amino acids that peptide spans between, so 572 and 585 in this case, and the position of the PTM. So this is at position 580 on the whole amino acid string, and it's a phosphoserine. We have the peptide form, so the notation where you can see that the modification is attached to the previous serine. The proteome it's mapped to, and the evidence. So that maps to the reanalyzed dataset. No, that maps the original dataset. Sorry, we haven't done the mapping to the reanalyzed dataset yet. And then under that, we have an additional section called PTM statistical attributes. Now, this is split between the datasets that provide the data for this peptide. So if there was multiple data sets that provided this peptide or, contained this peptide with this PTM, we'd have multiple PXDs here. And under each PXD, you'd have additional statistics and PubMed ID information and source information. That will very much depend on what data we've got from that data set, but we've included all relevant statistics that we can, basically. And our information help pages help explain them too. If you want a good example of this sort of data, that X session is quite good. So that's where we show that sort of data. We're going to flip to a live demo Colab notebook. We're going to give you the link after we've shown you the demo, so you can watch and learn, and then you can play at home. So we're going to swap screens. Do you want to steal my screen, Aurelian?
Yep, I'll do that now. Are you able to see the screen?
I am. Thank you.
Perfect. So yeah, thank you, Emily. So as you said, we're going to share the link to those examples later, so you can take those examples, play with them, and modify them as you need. So Emily showed a lot of data that you could explore on the website with the feature viewer. So clicking on those small features that you could see in the feature viewer, you will get the information, plenty of information there. But if you want to retrieve the information to use it maybe at scale, or if you want to, you wouldn't want probably to go click on all of them one by one and then copy all the information from the website if you need to analyze the whole protein, or if you need to analyze multiple proteins. So here, to do that, so as Emily showed, you could download the data, but you can also access the data and process it right away from the API. So the API is a programmatic interface to access that data. It is the same API that is used by the website. So whatever you see on the website, there is a way to retrieve that data programmatically. And so nothing is private. Everything that you see on the website, there should be a way somehow, somewhere to retrieve that data and process it. So here for the example, we used Python. This is a API that is language agnostic. So you don't really need to know Python to do that. If you prefer R, if you prefer another language, you can also access that data this way. There's no limitation to Python. We just need to have to make a choice for this presentation and to run this demo. And this is the choice that we've made. So here, just want to highlight two things. So first of all, the documentation. This is just in the comment here, but it's going to be useful. Every time you want to use the API, it's always good to have the documentation at hand. So we have the documentation published, and I'm going to show it in a minute. And then I'm going to store in that variable the root of that of our API, and then we're going to modify that URL to be able to access different data depending on what we need to do. And here it's just a helper function. No need to take too much to pay too much attention to that. It's just a helper to be able to do requests and return the information. So first, let's see an example. So we're gonna get HPP data, HPP peptides from a particular protein, so APP in that case. APP, the accession in UniProt KB for APP is P05067. So I'm going to keep that information here, accession. I'm going to keep that in the variable. And the URL that we'll try to access then is the proteins API URL that I've shown before. So that would be this URL. And to that, at the end, I will add the specific endpoint that I'm interested in. But Emily already highlighted in the presentation in the slides that the information is available at proteomics HPP endpoint. This is where you use the information, /proteomics/HPP. And in that case, adding the accession, which would be P05067 in that case. You don't need to get that. This is all in the documentation. So if you follow that link, you will get the documentation for proteins API. There's plenty of information. There's a whole section of proteomics, which is what we're focusing on today, but there's plenty of other information that you might be interested in at some point. This is the endpoint that we're going to be using today. So it says here that we need to replace this part with the accession that you're interested in. So that's why I'm replacing that with that variable in the code example. So I'm going to zoom in a bit, I think. I do it there. And the documentation also tells you what value will be returned. That's always useful because otherwise you get data, you don't really know what fields will be in there. So here you have an example value just with fake data, but to tell you what shape it is. So you will have an organism name, which will be a string. You will have a list of features and all of that. So that's a good way to have a look at what data you will get in there. There's plenty of data, and you might not be interested in everything. That's why you will process that data through programmatic access. You also have the model if you want to know. It's a different way of showing that. So if you're more comfortable exploring the model of data, this is the same thing. So it tells you, okay, the information will have this information. This will be a string. This, for example, might be optional. This will be a list of features and all of that. What is a feature? That would be this other object. So you will have this. Another way to do that is also, well, to just download the data and print it. So this is what I will be doing here. And so what I'm getting here is the huge object with all of that data. So yeah, there's a lot of it. So what I've seen before in the documentation is that the object that I will get has a list of features. So just to highlight how many features we have in there, I will print the number of features here. And so if I get the length of the features array in that object, I see that I have 980 features. Again, if you wanted to do that manually, that would be complicated. And if you want, that's where the programmatic access approach is interesting when you want to process these kind of things a bit more easily. So we're going to process this information and put it in a table. This is just a library I use there. You don't necessarily need to use that library, pretty table, but this is to have some kind of visual output. But you can output your data into a file. You can output your data into another process or pipeline that you have defined, or print it again. I don't know. We can do whatever you want with the data in the end. So what I will do, I will loop through all the features in my data. For each feature, I will have a look at each evidence. So I didn't invent that, it's in the documentation. So just bear with me, and then we're gonna have a look at the data in there. And then for each of the evidence, I will filter only the ones that are coming from HPP peptide atlas, because here, as I said before, I'm only interested in HPP peptide atlas data, just for this example. And then for each of them, I will also retrieve some properties that are specific to the source. So in that case, HPP peptide atlas will have a set of properties that are quite specific to it. That's also why I want to process them independently from other sources. And so in the table, I will extract some information like the the ID of the peptide, the start and end of the peptide on that entry, and the sequence, etc. So let's run the code and see what it does. Just additionally, I will want to sort the data. So in that case, I will sort by the column at index 1. So parametrically, indices start at 0. So column at index 1 will be the second column, and also by the third column. So here I'm sorting by where the peptide starts on the protein and also where it ends if they happen to start at the same place, like this case. And this is the data I have. So this is a subset of the data, as you see in the tooltips, and also in the dump of data I showed before. There's more data to it. There's links, there's cross references, and all of that. And here you can see that I have made a nice table with the information from P05067. You will see that on P05067, if I have a look at the position 9, this is where I will get this peptide. And some information, in that case, they are coming from the properties that I've extracted from the HPP atlas data. And it provides us with number of samples and number of observations. There was way more data, but just for the table, I decided to only print the 25 first record, but you can do whatever you want with the whole data. And also you can look through multiple entries if you have multiple entries and just collect them chronologically. Then I will do the same thing, but having a look at massive data. So HPP massive, this is another source. So the code is basically the same as I just showed before, but here the condition is to filter only the ones that are coming from this source. So if I run the code in the same way, there's nothing different from the rest, except from the properties, where the properties are a bit different because they're coming from this different project that provides another set of metrics. So here in that case, I will get also information of start and end, some IDs, and the peptide. And the things that are specific to massive are number of datasets and number of PSMs. So again, I can print those in that table. Okay. What proteins do my peptides belong to? So take that the other way around. You have a peptide from an organism, and you want to see which proteins in UniProt KB this peptide is found in. So in that case, you don't have the accession, but you want to find all the accessions that correspond to that. So you have another endpoint also documented in there. So it would be here. So what you can request, sorry, not that one. Let's see, yes, that one. You can see that I can query by different things. So in that case, I will query by peptide. So I have some peptides I want to query. So if I come back to my example code, I will see that I will pass the peptide I want to query. So these two peptides here. Also, I want to filter by the organism. Otherwise, it will search in all of UniProt KB. So you might return other organisms that happen to have the same peptide. It's not the case here, but it's always good to filter by the taxonomy that you're interested in that case, human. And then same thing, we'll create a table and we will put data from the API. So here, my data will have a list of entries. And for each of these entries, so that would be the entries, the UniProt KB entries that match these, that have this peptide somewhere. And then for each of them, again, I will have a look at the evidence and extract the different properties from that, and print this information in that table. So here, what I decided to print is the accession. So Q9BXU2. So if I go to the UniProt website and search for this accession, I will see the full entry. And if you go to the feature viewer, you will see the different peptides in there, one of which should include this peptide that is the one that I was looking for here. And where is it in the protein? It will be at position 187. You will also have the peptide ID here and the source. And same thing, I have another match here. I know that it's not matching to that peptide, it's matching to this other one. It's always good to, especially if you use multiple peptides, to check which peptide is matched again, because otherwise you will get a lot of results, so you might not know which peptide is matched again. So in that case, one peptide matched to one entry, and one other peptide matched to another entry. Yes, I think that's about it for the example. So we'll go back to the presentation. Emily, if you want to take back the screen, and I will share now the link with you to this to these examples.
There we go. Thank you very much. So if you want to scan the QR code and have a look at the Colab, you're going to get these slides after the webinar, so you can use the URL. That was just a brief introduction, and there are plenty of things you can do with our Proteins API. There is so much more proteomics data that we have. You know, every single one of those peptides has a URL, has a set of statistics, has a set of dataset IDs. It obviously depends on what data we get from those sources, but you know, the data is there. If you like accessing via programmatic methods, our documentation will allow you to do that with whatever language you want to use. If you're a Python fanatic, we actually did a programmatic access to UniProt using Python in the autumn. So if you look at the EBI training pages, we have a webinar that's recorded there, and it gives you a lot more examples of how to access different types of data in UniProt, not just proteomics stuff as well as some of our tools, I believe. So we're nearing the end. I'd like to thank our funders, which is primarily the NIH, EMBL, Open Targets, and CERI. We're a rather large group from all around the world that does curation, development, web development. We have a whole production team that helps us produce our data.
I think with this, we can move on to the questions. So we have a couple questions in the Q&A. So there is one which is a little bit unclear. So we'll start with this, so in case the person will have time to clarify. So the question is, can you repeat the process after Google Colab? So I'm not sure if it's just how to access it, or, you know, so maybe if you can just clarify the question, when in the meantime we can take the others. So we have one question.
You need a Google account to access this. I think it's worth noting. You log in, you click the link, you log in with Google, and you should be able to view it. And then if you get back to us, we can answer you another question.
Perfect, thank you. So the next one is, can you please explain an effective way to batch retrieve PTM information for a list of proteins?
Aurelian, that's you. Do you want to take that one?
Yeah. So yeah, you have a list of protein. In that case, you would do. Sorry, I'll share my screen again. Just a moment. Okay, so PTM information, but for a list of. So what we were showing information is for a given accession. So you have P05067, and then you access the proteomics information through those endpoints in there. Sorry, first I need to jump to the right section. That would be.
That would be the right section, depending on what they're searching for, I guess. So you can look in the non-PTM section or the HPP section for standard peptides. If you're looking for modified peptides, you want PTM.
Exactly.
But you can pull back similar from each endpoint, I believe.
You have two endpoints, yes, for those. That's the one I was missing in the middle is this one that I didn't see. And so here for that, you can, using that one without anything afterwards, you can do a question mark accession equal and then pass a list of accession. And so that would be up to 100. So in one query, or otherwise, otherwise you can use the same as I showed before. So passing the accession in the URL, so modifying the URL to include the accession like P05067, but then you would need to do that multiple times, one after the other. Just make sure that you don't launch like 1,000 at the same time. Just if you do in a loop, you request for one, you process it, and then you request for the other. Don't start sending thousands of queries at the exact same time. Otherwise, you will get blocked at some point. So yeah, The best approach would be this approach of querying for a list of accession, and then you will get a list instead of having the object directly that I was showing here with the list of features, you will get a list of entries, and for each entries, you will get a list of features. So that would be the way to access that. And I don't know if there's another problematic if since I still have the screen sharing, and if I have a list of peptide, I want to know the protein. So that would be the same thing as I showed at the end. So this is the other way around. You have this, you query for a list of peptides. Here in that case, I will pass all the different peptides I want in an array separated by, well, in the list separated by comma. If I want multiple of them, then it depends on your programming language that you're using. You can just add them. And I'm guessing there's a limit to do that. So it will be documented. I'm guessing 100 or so. So if you need more than that, you will need to do by batch. So you request 100 maximum, you process your data, and then you request the next batch of 100. Yes, that would be it for that.
And as you're doing this, especially when you're looking for peptides, just from a biologist perspective, Aurelian, just remember which endpoint you're calling from. So like non-PTM, you're calling from public data like proteomics-DB, peptide atlas. Think of the pipelines I showed you in the earlier slides. If you're calling from HPP, you're just calling HPP data. Okay, so think about the search space you want to use for these peptides.
Yeah, those three here.
Yeah, so those three are the ones we talked through today, and they've all got different inputs for UniProt. And obviously, if you're searching from UniProt, pulling that data from them, just remember the search space you want to look for.
And it's detailed, the origin of it a bit higher. Also, you can come back to this presentation if you need to, or explore the help pages in the UniProt website, and you will get more information.
Great. Thank you. So the person from before clarified the question. So they were referring to the dataset of APP. Which website did you use? The moment of Google Colab?
So we're calling APP from UniProt. So we're using APP as the gene name, and we're calling the accession. The accession ID is a UniProt accession ID, and we're calling that from. We're then calling the data from UniProt. So if you don't know the accession ID, I mean, we use an accession ID. You could search by a gene name and taxonomy ID if you want, but this is where
if you're on the website,
yeah,
you look for APP, you will get a lot of information. It would be better to search for your specific gene, and one of them will be the human one that you're interested in. But yeah, if you go directly to P05067, this is the UniProt-KB accession. This is the UniProt-KB ID, and this is the gene. So that would be something that might be in multiple accessions. And again, if you have a look at the feature viewer, this is the same data as we're using from the API. There's a lot of it in there, but if you look at proteomics, as Emily said, proteomics, and then I think that would be the ones I've shown were the unique peptides. So those, I don't know how many.
They're from the Human Proteome Project.
Oh, right.
They're within the Human Proteome Project, yeah. So they're
all those small rectangles, yeah, that would be the ones that you are accessing through programmatically. So you see the point of accessing that programmatically is that you don't need to go zoom and explore all of this in there.
And it just shows you all the different, and them tracks are a really good example of all the different types of data and the coverage different endpoints give you over a protein. So it might be that you search all three spaces as part of, depending on what you're looking at. But yeah, bear that in mind, which kind of endpoint you're looking at.
Okay, so we have another question. So can you please explain how can UniProt annotations help distinguish between protein isoform in proteomics experiments?
When you say protein isoforms in a proteomics group, it could get highly entertaining. So depends what you mean. So we do map unique peptides that only map to isoforms and only map to our canonical sequences. Our feature viewer that Aurelian just showed is our canonical sequence, right? If it maps to an isoform, you have to put the accession ID in for the isoform to see the data. The best way to view this data would be programmatically, Okay. So an isoform, when I'm talking about a sequence isoform here, i.e. when you read a paper, it goes isoform 1 is longer than isoform 2, like a splice isoform. Access that programmatically and pull it out using the accessions for the isoforms, and then you'll see your peptides. That's the best way to view that. With regards to a proteomics isoform, which could mean anything with regards to differential peptides, modifications, etc., that's what we're trying to do. We're trying to map as much PTM data as we can and the different modifications of this 3D proteomic structure. To clarify, were they asking about how to look at that in a large view? What was their last point there? How can UniProt help us?
It was like to distinguish between protein isoform and proteomics experiments. So maybe if the person wants to.
Yeah, that's more of a data set basis.
Yeah.
We're trying to bring this in large scale. We'd love to be able to do that. And if someone wants to look at our PTM data sets that are available publicly for download and start doing contrasting analysis between the different isoforms in those data sets, please do and let us know. That'd be great. I'd help.
Thank you, Emily. So we have another question. So there's one person asking, if I change the peptide according to my need, the results are not showing. I don't know if it was trying to do like programmatic access. Can it not show the results for novel peptide? So I'm not sure of this.
So
You want to take it? I mean,
I knew you were going to let me do it. So the endpoints we've told you today specifically look at proteomics peptides. So you're looking at tryptic peptides and different digestions that go through a mass spec. They fly, they get detected. We import them from repositories. So these are peptides that have already been seen. So essentially, you search UniProt programmatically, or you're looking at a protein entry, and you're already searching the data space. That peptide has already been seen by a data set that exists, and we've even brought it in. If you want to search the proteome for a peptide, or even a string of amino acids in a particular sequence, and you want the match for that, that is our peptide search tool on our website. That is a different tool. You can access that programmatically as well.
You can find that on the UniProt website. At the top, you have a list of tools. So blast, align, peptide search, ID mapping. So that would be peptide search. That would be any subsequent search within all the UniProt KB entries. So not necessarily a peptide as we know or understand as proteomics, but any part of a sequence that we have in there. So it's wider, but it doesn't guarantee that it matches a known peptide. So if you have a novel peptide, that might be a good use case for that. And also, if you don't find it there either, you could also do a blast and then restrict to 100% identity or really close identity. I don't know if that might be interesting for your use case, because maybe there's one amino acid that is different or whatever. I don't know. But you can blast is a sequence similarity, and peptide search would be a sequence exact match of part of the full or the full entries. I think that's it for this question. Is there another one?
Someone made a comment.
Yes.
While you guys look at that, someone made a comment about the statistics and that confidence scoring is important. Yes, it is important. The confidence scoring we're talking about with regards to, hang on a minute. Let me get rid of the Zoom bar because that's in my way. So when I say gold, silver, bronze, this is confidence scoring we've done with the University of Liverpool. This is novel. We have a publication out on this at the moment. Please see our help pages. If you click on the eye, you'll see the help pages. We have a whole method for how this gold, silver, bronze is done. It's novel. It's unique to the PTM exchange. When we're talking about statistics and false localization statistics, we have them too. They're completely separate from what we did with PTM exchange. So if you click your peptide, and again, accessible programmatically too, but if you click in your PTM containing peptide section, and you click your peptide you're interested in, and you go down to the statistics set, this is all the data that we've got statistically. I think we've even got a little bit more programmatically, actually. It's divided into the original data sets that are analyzed by the PTM Exchange project. So it's the IDs, And under each dataset ID, it's the residue number, the modification, the paper that's associated with if they published, the confidence scoring we did, and then where it came from, if we've got that data. We've got the PSM count, and then we've also got final site probability. And if you want to go even more in depth and you're a real proteomics nerd, you can see the USI. So you can go and see the actual spectrum that came off that mass spec and go and have a look at it for yourself. If there's multiple data sets that provide a peptide, I'd love to be able to find one for you. Here we go. So this peptide is seen in 3 different data sets. We have the statistics for all 3 data sets and all the USIs for that PSM in that data set. So please go and explore programmatically. There's even a little bit more you can download as well. So, yeah.
Great. We currently have still 3 questions, so I think we will have time to take all of them. So there is one person that is asking if there is a way to generate a detailed protein profile in terms of structure by using sequence with a mutation, if there is any tool on UniProt for this.
So
Not UniProt, I believe. No.
By using mutations, yeah, no, that would be, I mean, you could do that yourself by using the data chromatically. I mean, the feature viewer is an example of putting in correlation. If you have a look at the bottom of the page, you have structural information for the sequence. So that would be PDB entries or AlphaFold. And so if you click on a specific peptide in the track, you will highlight on the structure where that is. But if you want to analyze, if Emily, you want to show that, yes.
Well, I've zoomed in. There we go. So yeah, I've got a peptide that I've clicked here, and that peptide. This isn't a very nice structure. I apologize. I didn't pick. Oh, there we go.
You do have sections with a higher PLDDT. That might be interesting, but yeah, you can view all of that, or also in the track up there. So if you want to choose one that would be at the position where you have blue in the AlphaFold, that might be more interesting. There you go.
There we go.
And so here is where you can view this kind of information together, but we don't have mutation. We cannot analyze mutation directly in there or effect of mutation. I believe that might be where protvar might be useful. So there's another website in the ecosystem of UniProt that has this kind of variation information, and that is highlighting on structures or part of the structure where the effect of a variation might be happening. So you would be looking for something called protvar, PROTVAR. Yeah, we have links of that in the variant viewer section of each entries also. Okay, you have a good example? Yeah.
I always have a good example. I need to be there. The webinar on the 10th of April will touch on this, all the variants and where to find all the variant information, where we get it from, and there is a fair amount of modeling that goes off here as well. So if you click on, so this is the table. I don't want to explain all of this, but essentially every single dot here is a variant, and we have a table underneath that corresponds to said variants. If you click on the amino acid change, it takes you to protvar, and that details the coordinate, the genomic coordinates, the gene, the codon, the strand, the isoform, protein name, the change, the amino acid, consequence.
If you click on the detail, the tree structure, you will see that tree structure information.
There you go. Yeah.
Well, that's a bit of a teaser of the other webinar. So yeah, attend that one too.
We have it. It's just finding it. It's just knowing where it is.
Okay.
Great. Okay, we can move on, I think, to the next question, which is if there are any recommended approaches for mapping peptide identification to UniProt entries.
So specific IDs of a peptide that we know. So to look for peptides, we highlighted already. So the search for specific peptides, as in my example. Otherwise, you have the website. If you cannot find it, you have the peptide search that would look for any subsequence. But if you have IDs, let me just find that might be one of the parameters.
If you're searching raw peptides, as they are coming off a mass spec, you want to be searching against a reference proteome.
Also, yeah.
Yeah.
But I'm wondering if you come with an ID that you already know and you want to find it in our data. It might be a way to do that. UPID, so that would be proteome ID plus Data source. No, okay. No, you would need to search by the peptide itself, not by the ID of it. That would be the best way.
And again, remember your search space. Do you want to know if your peptide of interest has already been seen as a peptide, or do you want to know whether that matches a proteome and a protein in that proteome? So you either want to use one of the proteomics endpoints we've talked about today, or you want to download the whole proteome and search against that as though you would doing a proteomics experiment off a mass spec.
Okay, then we have, I believe, a similar question because someone is asking if I have a table with my proteomic results and I have an UniProt accession number and a tremble, do I count the protein as one protein or two separate ones?
Okay.
And that depends on your analysis. I've had them tables.
Also, just to clarify, I mean, UniProt is the website. If the accession that we're using are UniProt KB accessions. So if it's in the same table, you have two accessions from UniProt and one from Trumbo. You have UniProt KB, and within UniProt KB, you have SwissProt and Trumbo. So Trumbo is also UniProt KB. So basically, they would be, if they appear in the same table and you recognize that one is from Trumbo and the other one is not from Trumbo, they're still part of the same data set. The only thing is that the one that is not Trumbo is SwissProt, so that would have been reviewed by curators. Trumbo would have been annotated automatically, but they're part of the same data set. So they might be two different proteins, or they might be the same one if that's not been integrated yet. I guess then that once the clarification about what is UniProt KB, what is SwissProt, and where Trumbo is done, then that's more of a curator question of like, are they the same isoforms or something like that, Emily? That might be the case or not?
It depends on your research question, right? As I say, if you're mapping to SwissProt and you're only looking at reviewed entries and you're just getting SwissProt entries back, you're looking at the entries that are manually annotated by curators, and that means you've got all the annotations are checked manually by someone in that field or expert in that pathway or that family of proteins. So every single one of us curators specialize in a particular area. That means you've got data from publications and studies that are out there, in addition to everything that we automatically bring in as cross-references and automatically annotate. Having said that, even a protein such as beta-catenin that I've just shown you, or GSK3-beta, which are big proteins that people know, you've still got tremble entries that are associated with them. And that means we've had sequences brought in that, they might even be identical to the SwissProt entry, and that's why your peptide matches. They might be slightly different. They might be an isoform that we've not got around to curating into the SwissProt entry yet, so that might be valid. It depends on your experimental question. If you're just saying, just identify my proteins, you could probably search SwissProt. If you want to say, I want to know all proteins, all isoforms, regardless of annotation, pull them all out and have a look. Eventually, you're going to have to pick your favorite protein and manually have a look at where your peptide matches and why it's bringing a tremble entry out and not a SwissProt entry, as an example, which is easier in some organisms than others, because obviously, like human, we've tried to manually annotate everything that we think is a protein at existence level. It is less so for mouse and less so for rat, as an example, going down the model organisms. So it's very much going to depend on your experimental case and what your question is, and you should know that before you put your data in your mass spec. So go back to your hypothesis and see what you're really asking.